I'd like to explain a little bit of our work that we did together with many partners in the field of modeling and simulation molecular communications in flow. Molecular communication as an alternative to radio, that is something we are all interested in, and that is why we are here to exchange ideas, to exchange state-of-the-art. And this actually started many years back, right? Early papers have been written something like almost 20 years ago in the field of bio-inspired networking. Then on the more, let's say, molecular communication side, introducing the Internet of Nano Things, papers dating back to 2010. At that point in time, we started working in this field 2015 with connecting in-body nano communications with body area networks. So we have as a research community have quite some experience in the field already. So when we talk about molecular communications, we are fortunately now moving from this stage, having a purely theoretic concept and moving towards more realistic applications with lots of experimental setups. So Robert explained some setup. Tuna explained some setup that we use for experimentation in the field, and many things are now getting more clear, right? Our theoretical models the gaps in our theoretical models are being filled with new experimental insights, with new empirical evidence that we gain from many of these experiments. But at the same time, we observe that some wet lab experiments that we can do are already getting us lots of insights, but still are limited in size, limited in scope, and possibly we can do something in a larger scale. So we are particularly interested for the presentation today in the field of what is called macroscopic molecular communications. So looking at a very macroscopic level and how to eventually study applications. Now, when I talk about macroscopic molecular communications, then I'm very much interested in not just a single molecule or a group of few molecules that are used for modulating some information and getting some information so from A to B, but trying to do that large scale, trying to do that in medicine, for example, in the entire human circulatory system. So for measuring, let's say, observations from biomarkers that we can observe, or in industrial environments also, when we consider large chemical plants, then these plants are very hard for radio communications, and maybe we can do a lot of communication among nano devices using molecular communications. So for the presentation today, I want to introduce some simulation framework that we have been working on. And as for many of us, everything started with an experiment. And this experiment was actually done in Erlangen. So that's a collaboration in a project that we also did together with Robert Schober and many of his colleagues in Erlangen. And the experiment looks like the following. So we have a water-based liquid environment in a tube where we have flowing liquids. So that is coming from the left side here with continuous pump being moved to the right side to this waste container. And we are able to inject molecules, in this case, magnetic nanoparticles, that we can magnetize on demand into this flow, and we can later on, at a later stage, a different position, measure whether we observe some of these nanomolecules by having a detector, in this case just an inductive detector for magnetic particles. So far, it looks very, very abstract, maybe. This is the real experiment being executed in Erlangen. And here you see on the right-hand side the sender system. So that is a pump injecting magnetized particles into a flow of water. That is the flow of water that you can observe here. And we can later on measure the injected particles using this inductometer, so measuring induction of the reductive permissibility of the liquid that we observe. I have a quick video showing the experiment. As you can see, the nanoparticles are colored in black, so you can even visually see the flow of these particles in this environment. What you can do now, well, doing classical telecommunications, right? We can modulate some information on top of these particles. The results presented here are actually for different type of particles, published just very recently in 2021 at Nanocom, and you can see on the left-hand side channel impulse response for some of these particles. On the right-hand side, you see even a sequence of transmitted bits. Actually, it's 80 bits that are currently transmitted. And fortunately, already visually, you can observe the communications here. And the group in Erlangen is currently working on increasing capacity for the communication on this flow experiment. Now, this is a small scale experiment, right? This is definitely not large scale. This is definitely not covering all our human body. This is definitely not covering a large scale chemical plant. So we tried, starting with this experiment, modeling the system in a simulation environment so that we can finally experiment on the large scale. So when we look at existing models, there are actually two extremes, let's say, available in our scientific community. Either we do a very theoretical model using transfer functions and statistics, which is not very accurate and not very representative for individual effects, or we do the very, very, very complex multiphysics simulation of particles, like in other communities that are investigating very, very fine-grained microscopic effects in liquid environments, in fluid environments. So we tried to find some sweet point in between, not getting too complex, but still being able to model the effects rather accurately, so that we can study on a large scale microscopic effects of molecular communications. So what we did is we modeled all the pipe system in our system. We started modeling the flow in our pipe system, so that we can identify different flow characteristics, different flow properties within our pipe system. So typically, for example, the flow in the middle, in the center of the pipe is much faster, much stronger compared to the flow at the edges of the pipe, so that we can later on use this simulator to get exactly the same digital communications using molecules. A little bit more details, so that you can follow the general concepts and ideas much better. So we started modeling the pipe essentially as a 3D modeled, let's say, container. Within the pipe, we added flow using classical fluid dynamics. So you see, according to the arrows in the pipe here, that these are vectors, and the vectors represent in length and width the characteristics of the flow in a particular area of the pipe. So in the middle of the pipe, certainly longer arrows, because there is a much higher speed compared to the edges. We added sender and receiver system to our pipe system. So a sender system is essentially a system that can artificially release particles at different areas of our pipe system, so either in the center or more at the edge side, and also according to time characteristics, having a release of particles over time according to our transmission system. And we have a receiver. The receiver is essentially a counter. Now you count particles at the receiver system. Again, counting allows you to identify particles, number of particles that are more center or more at the edge of the pipe. But in the end, it's a counter, and with this counter, you can later on identify whether you are able to reach a certain signal threshold, so signal level in our system. So if you have all that available, we can start releasing particles, and these particles move according to the predefined flow, and the predefined flow is exactly the arrows in our pipe, and we can, in a time step based, discrete event simulation base, move the particles according to the flow dynamics inside the tube system. And if we can do such simple, we can do way more complex environments. So you see an example here, right? We have a more complex flow system containing multiple sectors of pipes. The pipes may be connected to pumps that introduce either flow or particles. We can have sensors measuring the number of particles at a certain area. And this scenario can get almost arbitrarily complex. Certainly with complexity, the computational effort is also increasing that you need in order to evaluate the particles. So a little bit more about tooling. So all the 3D models are generated in the tool Blender. So it's a very popular 3D modeling tool, which also allows later on doing visualization of the system while flow is in action. So we model all the pipe systems. Flow is automatically generated in the background according to fluid dynamics calculations, and the release of the particles is again can be visualized in the scope of this Blender tool, which allows us nice animations. So this is an example, very simple example certainly. And in this simple example, you see on the left-hand side a pump that is releasing particles. On the right-hand side, you have a receiver system, and this receiver system later on does the counting. Let's have the particle go. You see in the middle of the core of the pipe, particles are moving much faster compared to the edge of the pipe, and you see at the right-hand side, the color is changing, and this is exactly where the particles are being counted in order to identify communications. So using this simulator, we can represent 100% accuracy the experiment that I mentioned in the very beginning. So we started with a simple experiment that you can see here using on-off keying modulated transmissions. And what you see here is we measure the susceptibility, so the number of particles that are containing some magnetization in our flow system, and we even send some modulated information by artificially releasing particles according to this on-off keying. We can play around with different modulation schemes now, which is very straightforward. And you see that we can even do more complex modulation schemes, certainly having an influence according to how short we can do symbols and what is the resulting bit error rate, which is very easy to calculate in this simulator. So the resulting system was published in 2021 in IEEE Transactions on Molecular, Biological, and Multiscale Communications, and is now available as open source for our community for doing your own experiments. So quick intermediate conclusion. So what we have available is we want to study macroscopic molecular communications. First wet labs experiments are successful, but scale is not yet at the dimensions that we are looking for. So our simulator, I forgot mentioning that the simulator has a name, right? It's called Pogona. So the simulator allows doing much larger scale experiments, and it is 100% validated against experimental data. So from here, we looked, well, if we can do this type of a communication, of molecular communication in a liquid environment, why not trying the same thing also in air-based molecular communications? In the end, we thought, well, conceptually, in air-based environments, we also have a flow, and molecules move mainly according to flow. In liquid environments, there's also diffusion, so they move a little bit according to diffusion, but the main influencing factor is flow. In air-based environments, this is slightly different. In air-based environments, flow has a huge impact, but also gravity has a second dimension, and this gravity has a very strong impact as well. So we started again with an experiment, this time with some colleagues in Kiel University in Germany, and the experiment looks as shown here on the slide. We have a large container in which we are able to inject particles on the left-hand side, and we have kind of a spray nozzle on the left-hand side, and the intensity of this sprayer gives us the flow characteristics. And in addition, we have in this container the ability to observe the movement of our particles, because we use illuminated particles that can be illuminated using ultraviolet flashlights, so that we can exactly follow the trajectories of these particles, and later on represent the same system in a simulated environment. So I have a video. Let's hope I can get the video running here smoothly. On the left-hand side, there is this testbed environment, and you see the particles in this illuminated fashion coming from the right to the left in this case. And you see most of the very fast particles are moving horizontal line, but the slower particles also have a tendency going towards the bottom of this testbed container because of gravity. On the right-hand side, we have a simulation of the same effects. It's now from left to right, but otherwise it's the same thing. We have a sprayer again. The sprayer induces these particles. We use Pogona again as a simulator for simulating flow, but the flow is now overlaid in addition with a second vector of dynamics of our particles, and that is gravity. And eventually we get exactly the same results in our experimental setup, like in the simulator, right? So I hope you got a little bit interested. So let me make a projection, right? And this projection is molecular communication at some point in time will become a part of all our communications environment, possibly, but certainly not in 6G, but at some point in time it will become part of our communications environment. And I am very interested in looking into molecular communications in a much larger scale, in fluid environments, in air-based environments, and particularly also within the human body. So we are currently doing a project together with some colleagues in Lübeck, in which we model the complete circulatory system of a human body. So all the arteries and capillaries of our circulatory system, so that we can simulate not just artificial pipe systems, but we can simulate the blood flow in a human body, allowing us to get more insights on the movement of nanoparticles. These nanoparticles can be even nanosensors, but maybe just molecules within the human body, and we can derive some information of how long does it take from observing some biomarkers in the human body to finally deliver these observations to an out-of-body gateway system, so that we can observe some impacts on what is the communication probability, what is the success rate, what is the time it takes from measurement to final observations. So lots of new ideas out there, and I'm very happy and very proud that we can do this work together with many colleagues, both from collaboration partners, but also within the group here in Berlin. So I definitely want to highlight Jorge Torres Gomez working with us on these systems. Lukas Stratmann was behind the development of this Pogona simulator. We have collaboration in Erlangen, mainly with Robert Schober and Harald Unterweger, Jens Kircher, Max Bartonik, helping us with on the experimental side. We have the collaboration in University of Kiel, led by Peter Höher, and the PhD students were Martin Damrat and Sunashir Badachayi. And we have collaboration in the University of Lübeck. Head of the group is Stefan Fischer, and the cardiovascular system simulation is mainly done together with Regine Wendt, and of course with many others in the field. So with that, I'd like to conclude. Thank you very much.